So, good afternoon, everybody.
Good afternoon.
Afternoon. Today I want to finish the genitalia system with the placenta. We have 2 slides only to demonstrate the properly matured placenta and the 6-week-old placenta. And after that, we can do a little review from the important topics of the histology. Now let's do that. No. So firstly, let's see the 6-week-old placenta. So we can see only the chorionic villi here. And you know, when we're talking about the placenta, that is eventually a discoid organ, a discoid structure where the gas exchange occurs in the fetal life, during the fetal life, is located in the internal surface, the middle superior internal surface of the uterus. And the placenta is eventually just a spongy-like structure having villi, having finger-like processes. Now we are looking at the tips of these chorionic villi, and eventually these structures are growing from the extraembryonic mesoderm from the fetal side, and among the villi we can find blood, the maternal blood. And the gas exchange occurs by diffusion through the villi wall. When we're talking about villi, we can distinguish 3 types of villi, chorionic villi. We have the primary, secondary, and tertiary villi. Primary villi just build up by trophoblast cells. So only trophoblast cells build up the villi. Secondary villi, but we can see here now, these are the typical appearance of secondary villi. Here the villi represent mesenchymal core. So there is a mesenchymal core having typical mesenchymal cells. You remember these are sometimes triangular shaped, having long processes. So these are typical mesenchymal cells. So when we have trophoblast cells externally and the core is formed by mesenchymal cells, that is a secondary villi. But eventually, in the really early phase of the development, we can find some tertiary villi, and eventually from the second, third months, only tertiary villi are presented in the placenta. That means in the mesenchymal core we can find some fetal vessels. So tertiary villi represent fetal vessels inside in the mesenchymal core and surrounded by trophoblast cells. So when we are looking at a bigger villi, for example, here or maybe there, so here we have some capillaries. Here we have some capillaries. These kind of villi are tertiary villi. Now let's see what we can see now. The outermost layer is called syncytiotrophoblast. That's the syncytiotrophoblast layer. Syncytiotrophoblast, that's the syncytiotrophoblast layer, that's the outermost one. And underneath the syncytiotrophoblast, so the closer to the mesenchymal core, we can find a cytotrophoblast. So cytotrophoblast layer. So syncytiotrophoblast, that's the outermost, and cytotrophoblast layers form the inner one. And eventually, we should mention that approximately at the end of the 4th month of the pregnancy, the cytotrophoblast cells reduced and only the syncytiotrophoblast covered the villi. So after the 4th month, the end of 4th month, syncytiotrophoblast layer covers the villi. Okay. So there, again, underneath the cytotrophoblast layer here, we can find the basement membrane, the lamina basalis here, where the cytotrophoblast cells just resting on. And underneath them, we can find the mesenchymal cells, Now let's see these vessels. Maybe that is really nice. It's a proper cross section. So here we have the endothelial cells. Here we have the lamina basalis of the endothelial cells. And inside we can find some cells. But they are a little bit weird. We never seen before that, these kind of cells. These are red blood cells, but this is a really weird appearance of the red blood cells. You know, the matured red blood cells don't represent nuclei, but we are at the very beginning of the development, and the early red blood cells, the developing red blood cells, represent nuclei. So the nucleated red blood cells are the typical structures in the vascular capillaries. So these are also, so here we have endothelial cells, just indicating the vascular capillaries, and we have nucleated red blood cells in the developing placenta. This is a 6-week-old placenta. So again, here we can see the trophoblast layers. That's the syncytiotrophoblasts. It's a kind of cuboidal or tall cuboidal layer. There is a normal cuboidal syncytiotrophoblast layer. Underneath them, we have the lamina basalis. Here we have the mesenchymal cells, the mesenchymal core, but it's a really nice, there is a longitudinal section of a capillary. And then this is the lamina basalis for the endothelial cells, and here are the nucleated red blood cells. That's the place called intervillus space. Intervillus space. Intervillus space for the maternal blood. So the maternal blood fills the intervillous space up. So, and I said the diffusion, the gas exchange occurs by diffusion through the wall of the chorionic villi. So you should know the components, the layers of the blood placental barrier. So the first one, the trophoblast cells. So trophoblast cells, you know, we can mention the eventually both at the first part of the pregnancy and at the end of the 4th month, when we have cytotrophoblast layer as well, but later just the syncytial trophoblast we have. So trophoblast cells. Then the number 2, the second layer, is the lamina basalis of the trophoblast cells. That's the second layer. The third layer here, the mesenchymal cells. So mesenchyme. Mesenchyme, and that's the third layer. Then we have the lamina basalis of the capillaries. That's the 4. Endothelial cells. And the last layer is the endothelial covering. So especially the tight junctions connecting the endothelial cells to each other. So that these are the layers of the blood placental barrier, or also called the layers for the diffusion from the intervillous space to the fetal blood circulation. So that's extremely important, related to the placenta. Yeah. Let's move on. That was the 6-week-old placenta. Let's see the full-term placenta. That's the 85. That's the one. No. So it's a fully matured placenta having 2 sides. Here we have this side showing these big lumens. That's the fetal side. That's the fetal side. And here we have the maternal side. So maternal side connecting directly to the uterine wall, the endometrium. And that's the fetal side facing with the fetus. So, and here we have the meso of the villi just growing from the fetal side. Everything on the fetal side, everything, the villi, the vessels, the layers are derived from the extra-embryonic mesoderm, extra-embryonic mesoderm. Now let's see the surface, the fetal surface. First, we can find a simple cuboidal covering epithelium. So they are the eventually a special covering what we seen onto the surface of the cord, the umbilical cord, the amnionic layer. So the amnionic cells are covering the fetal cells, amnionic cells, you remember. Amnionic cells, and underneath the amnionic cells we can find the amnionic mesoderm, amniotic mesoderm, so this kind of connective tissue, but it's an artificial gap. It's thicker. That's the what I'm showing. So there. So this. Oh my God. So this is the amniotic mesoderm as well. That is an artificial gap. And then the next layer under it, representing these fetal vessels, the umbilical arteries, veins, so called this area completely called chorionic plate. This is the chorionic plate. So chorionic plate, that is the chorionic plate, having the fetal vessels, the umbilical arteries, veins, surrounded by chorionic mesoderm. So this connective tissue surrounding the vessels called chorionic mesoderm. So here we can see the vessels there, that they are typical vessels having endothelial cells, muscle layer, adventitia, nothing special. But this is the chorionic mesoderm. Don't forget, it's a developing tissue. So we have mainly mesenchymal cells and some connective tissue elements, but it's a developing body, so developing structure. So we have mainly mesenchymal cells. And from the chorionic plate, here, chorionic villi start to grow toward the maternal surface. So, and in the, so I'll just zoom out. So this area, this represent the cross sections and longitudinal and transverse sections of the chorionic villi. So the mass of the chorionic villi are presented growing from the fetal side. And I'm telling you again, everything, the amniotic layer, amniotic mesoderm, chorionic mesoderm, chorionic plate and villi are derived from the extra-embryonic mesoderm. Now let's see the villi here. You know, I told you we have 3 types of villi, and definitely in the full-term placenta, we have only tertiary villi, so the well-developed villi, having trophoblast layers, mesenchymal core, and fetal vessels. So I choose this one. Yeah. So this is a proper cross-section. So these are the syncytiotrophoblasts, you know, the cytotrophoblast reduced at the end of the 4th month. So syncytiotrophoblasts are presented. Somewhere in some villi, maybe some remnants of the cytotrophoblast cells are presented, but mainly syncytiotrophoblasts are covering the villi. Definitely underneath them we have a lamina basalis, and there is a mesenchymal core. Just a second, there is a really nice huge cells. There is a huge cell. It's different than the other mesenchymal cell. This is a mesenchymal, that is a mesenchymal, also mesenchymal cells, but it's completely different. This is a macrophage. This is a macrophage called Hofbauer cell. So Hofbauer cell or macrophage, it's a placental macrophage or Hofbauer cell. It's just eliminates the old and the degenerated connective tissue cells. So that's the one. So if you can find a huge cell in the mesenchyme, that is a macrophage. And then here we can see there is an endothelial cell which also rests on lamina basalis there. So this is the lamina basalis, endothelial cell, the nucleus of that, and in the fetal capillary we can find red blood cells, but they are matured red blood cells belonging to the fetal circulation. So no nucleated red blood cells anymore. And just want to show the difference. Eventually, there's no such a big difference, just the location of the red blood cells. So they are belonging to the fetal circulation, and intervillous space having the maternal blood represents the red blood cells of the mother. So these are the maternal blood, the space for the maternal blood. Red blood cells here and red blood cells there, just providing gas exchange through the layers of the villus. So syncytial trophoblasts, basement membrane, mesenchyme, lamina basalis of the endothelial cells, then endothelial cells themselves, and that's it. So the red blood cells can absorb and take it off the carbon dioxide. So can absorb the oxygen and take off the carbon dioxide. So here we have the exact layers of the blood placental barrier in the full-term placenta. Intervillus space for the maternal blood, and in the villus there is a circulation of the fetal blood. That's extremely important. Yes, and some of the villi, so here, represents this homogeneous pink material, especially really close to the maternal surface. So here, there. That's the fibrinoid. Fibrinoid. The fibrinoids are just presented because of the aging of the placenta. So eventually at the end of the third term, the last term, at the end of the pregnancy, the placenta start to degenerate, representing this fibrinoid, which eventually blocks the gas exchange through the wall, because eventually it's a kind of degeneration of the villi. And now we covered the placenta, especially the fetal side. Let's see the maternal side. But just before that, there is a long structure in the placenta here. That is an extremely long villus from the fetal side, and it reaches the maternal side. That's called anchoring villi. Anchoring villi. So these anchoring villi reach the maternal side. The rest of the villi, the majority of the villi, are free villi. So you know, the villi are eventually they look like a tree. There are long structures, long villi reaching the maternal side, and there are free villi, just they are the shorter ones. And on the maternal side, everything is derived from the decidua basalis. Decidua basalis. From the decidua basalis, therefore we can find here decidua cells. So we can find decidua cells, showing some. So these cells the small cells there, these, not the big ones here. So these are the decidua cells. There are others. So the small cells are decidua cells. Yeah, some of the small cells, but they are big ones. So not these big cells, these small ones, these. So the decidua cells, so they derive from the decidua basalis and represent some septa, dividing the maternal side into lobes, small lobes, because on the maternal side, when we cover the gross anatomy of the placenta in the dissection room, we can distinguish the two sides because it's approximately a 20 centimeters big structure, so it's a discoid structure, and on the maternal side we can find 15 to 20 lobes separated by the connective tissue septa. There also there are cellular elements, beautifully seen. Small cells and big cells. So these are the decidual cells again. These. And again, just looking at the maternal side as well. So syncytiotrophoblast, mesenchyme, endothelial cells, basement membrane of the endothelial cells. So this is the exact route for the gas exchange. Yeah. Now, so that was our last slide in this semester. Now let's see some important slides from the material. Start with this. So this is the intervertebral disc, you remember, which eventually shows a bony part. There's a spongy part of the bone having red bone marrow. This is a slide to show the exact sources of the platelets, the megakaryocytes, but also we can find some components of the intervertebral disc. So when we are looking at that area here, that is a really nice one. So the color is different. Really close to the bone marrow cavity here, we can find the Haversian systems, the osteons. That's the Haversian canal, and here we have the circumferential or special lamellae just surrounding it. So these are bony lacunae here, there, having osteocytes. So osteocytes. But look at this. It's a different, just the color, but here they are chondrocytes in their lacunae. They are chondrocytes here. By the way, you know, it's a specific cartilage related to the intervertebral disc. And usually the cells are located alone in this kind of holes, in this kind of lacunae, and called this type of cartilage. Here we have maybe two, but they are not eventually real chondrons, just chondrocytes in lacunae. So this is a fibrocartilage. This is a fibrocartilage tissue, and close to the fibrocartilage here, when we can see the connective tissue surrounding the intervertebral disc, we can see real connective tissue here. So here we have the fibrocytes, the typical appearance of the fibrocytes. These are coarse collagen bundles. So this is a typical fibrous structure having fibrocytes. They produce the collagen fibers. So this is a connective tissue, but this area here, these are chondrocytes in the fibrocartilage. Okay, so in the bone marrow, we have numerous elements of the erythropoiesis, red blood cells, everything, and we have bony structures as well. Oh, sorry, they are not. These are the bony structures, the eosinophil ones, and these paled ones are just the preservated plasm. And there are some multinucleated cells, this here. This is a megakaryocyte. This is a megakaryocyte, which is the eventually which forms the platelets, the thrombocytes. Then you know in the urinary system, the minor, major calyces, ureter, urinary bladder, lined by umbrella cells, lined by urothelium. The umbrella cells are dome-shaped cells, sometimes binucleated, and underneath them we have huge cells, the pear-shaped cells, and at the bottom of the basement membrane we have the basal cells. So it's a stratified layer, but it's specific for the urinary system. You know, we have some slides representing neither muscularis mucosae nor submucosa, such as the gallbladder, seminal vesicle, ureter, some part of the bladder, and fallopian tube as well. So looking at this section of the gallbladder, here we have the mucosal folds lined by simple columnar epithelium, and this simple columnar epithelium at the top represents the microvilli. It's harder to see, maybe there. Then that's the lamina propria, which is a cellular rich connective tissue, and underneath the lamina propria here, immediately the muscularis externa begins, having the smooth muscle fibers there, and under it there is a connective tissue. It depends on the exact location of the gallbladder. So because if we can find a really sharp covering externally, that is a serosal covering, you know, the gallbladder posteriorly represents peritoneum, therefore represents serosa, and that side which faces with the liver doesn't cover by peritoneum, so it represents adventitia. So now we are looking at the peritoneal covered side. So underneath we have a subserosa and connective tissue elements, but it's just eventually, just because of the preservation, there is a artificial big gap. And definitely we can find some infiltrated lymphoid infiltrations of the propria, and some glands are also presented here. The cuboidal cells are typical for the glands. Then also extremely important, it just covered by the first few weeks, the stratified keratinized and non-keratinized epithelium. So here we can see it's nice one. So the stratum basale, stratum spinosum, and you know the exact shape of these cells caused by the desmosomes, connecting the cells. And then here we have the granule accumulating cells. That's the stratum granulosum, representing keratohyalin granules. Over the stratum granulosum, there is a red, homogeneous red area. This is the first layer having no living cells. So just compressed dead cells are presented here. The stratum lucidum presented only in the thick skin. So the surface of the palm and sole. And over the stratum lucidum, there is a thickest layer, that's the stratum corneum. And now here we can see the exact cross-section of the, or sections of the spiral excretory duct of merocrine sweat glands. Under the epidermis, that's the epidermis, under it we have the connective tissue elements with coarse collagen bundles and fibrosites. So these are fibrosites, and here we have the collagen fibers. And in the dermis, beautifully seen, these units. These are merocrine sweat glands, merocrine sweat glands. These are the excretory portion, and these are the, sorry, I mixed. That's the duct, that's the secretory portion. So secretory, the paled one, and the two layers, the darkly stained, that's the duct. That's the excretory portion, secretory portion, secretory, and that's the excretory portion. Yeah, that's the same. So the secretory and excretory. That here, that's the transition between the secretory and excretory portion. That's the lumen of it. Yeah, and underneath here we can see many adipocytes. You know, the adipocytes, the white adipocytes contain only one fat droplet, which is dissolved during the routine histotechnics. So now we cannot see the fat itself. So just the dissolved fatty components and the space for it is presented. So, and the nuclei, in somewhere we can see the nuclei of the cells, yes. So here we have the nucleus of the adipocyte, and there is a thin rim of cytoplasm just pushed toward the cell membrane. So this is the place for the fat, but it's dissolved because of the ethanol of the preparation. In the esophagus, there is another type of epithelium. So stratum basale here, stratum spinosum or polygonale, and at the top the compressed layers. These are part of the stratum planocellulare, spinosum basale. So that's the stratified non-keratinized squamous epithelium, what we can see in the esophagus, oral cavity, surface of the tongue, vagina, so this area. Also, important part, the ossification. Now we are looking at the frontal section of the nasal cavity. So this is the nasal septum, that's the nasal cavity itself. Here we have the developing maxilla, and that's the cartilaginous part of the nasal septum. Everything is lined by pseudostratified columnar ciliated epithelium. So we have respiratory epithelium. And also we can see the external surface here. So these are the these are the hair follicles, and it's lined by stratified squamous keratinized. Maybe that is a little bit thicker. So it's a developing, so it's not, it's a little bit different than the previous one, but I think we have some parts which is quite similar. So what we can see, the stratum basale, stratum spinosum, and here we have we cannot see beautifully the corneum and the granulosum. But what we can see, these structures associated to the hair follicles, the holocrine glands. You remember the sebaceous glands secreting sebum. Now, let's see the ossification. You know it's membranous or intramembranous ossification, but we can see here. These are the spicules, the trabeculae of the bony tissue. And inside of the trabeculae, we can find osteocytes in their lacunae. So these are osteocytes. And onto the surface, we have osteoblasts. So on the surface, we have osteoblasts. These are, I try to find a sharp area. No, that's better. So these are osteoblasts there. And you see the osteoblasts just lay down this pink or pinkish material. That's the osteoid, the fresh, the newly synthesized bone tissue. And then this osteoid eventually encloses the osteoblasts, and it becomes osteocyte. So therefore, the trabecula, the spicule, is getting thicker and thicker. To keep up the balance between the synthesis and the absorption, we have another cell type. That's the osteoclast. Here are. This is a nice one. Also multinucleated cell there. With acidic hydrolysases, they dissolve, they absorb the bone tissue, forming the house-shaped lacuna. So there is the own lacuna for the osteoclast called house-shaped lacuna. But everywhere here we can find vessels and mesenchymal tissue. That's the other type of ossification. And there we can see that's the metatarsal. So you remember when we covered these topics, the main difference between the intramembranous and endochondral ossification, the exact framework. So the intramembranous starts with the mesenchymal cells carried by the bloodstream. So mesenchymal cells differentiate into osteoprogenitor cells, and those differentiate into osteoblasts, just laying down the osteoid, just forming an island of bone. That's the intramembranous. But here, the story starts differently. So the mesenchymal cells from the bloodstream first differentiate into chondroblasts, forming cartilage. First, the cartilage, it's just think about a long bone. You know, the metatarsal is a long bone. So there is a cartilage structure called cartilage matrix. And this cartilage, you know, supplied by the neighboring vessels via diffusion. So it eventually receives the gases and nutrients from the neighboring vessels. But the bone tissue is vascular. You know, just think about the Haversian and Folkman's canals. But no vessels in the cartilage. That's the point. So one is avascular, the other is vascular. The bone is vascular. Now, so we have a cartilage matrix. We have a cartilage matrix first. But later, when the development, when the time goes on, a bony collar, a bony cushion-like something, but called bony collar. A bony collar is start to develop at the mid portion of the cartilage. You know, I told you the cartilage receives nutrients from the neighboring vessels. So that collar eventually deprivates the mid portion of the cartilage from the nutrients, causing degeneration in the cartilaginous tissue. Therefore, we can distinguish different zones in the developing structures, in the developing bone. So that area which is far away from the bony collar, this is the bony collar, shows normal appearance of the cartilage. So we have typical hyaline cartilage with chondrons. It's a healthy appearance. Definitely they are smaller than the adult, the matured hyaline cartilage chondrons, but it's quite similar to them. So that's the resting zone. That's the resting zone. And then a little bit closer to the bony collar there, the cells, so when the process eventually effects on this area, so the chondrons and the chondrocytes are proliferating, raising their numbers. So that's the proliferative phase. That's the second phase, just closer to the bony collar, because the chondrons feel the less of nutrients and less of gases, the oxygen. So that's the proliferative phase. And then more closer to the bony collar, they are properly degenerating. So here they are getting bigger and bigger, the nuclei are getting smaller. So the typical signs of the degeneration are presented. They become hypertrophic. So this is the zone of degeneration or zone of hypertrophy. And there, so almost at the level of the edge of the bony collar, where the cartilage doesn't get any kind of vessel, the cartilage matrix breaks down. So here we have these are part of the degenerating cartilage. That I can see they are dying cells. So look at that here as well. So they're completely destroyed by the deprivation of gases. And when we just zoom in here, this area, this is a best area. You know, the cartilage matrix breaks down. So because of the deprivation of nutrients, forming large cavities. These are. And with the vessels penetrating the bony wall, because it's quite normal, because it's a vascular tissue, the penetrating vessels, mesenchymal cells, enter into the bony cavity, the bone marrow cavity, and they differentiate into osteoblasts, just lying onto the surface of the cartilage. So look at that. Here we have, this is a cartilage matrix, and onto the surface we can find these kind of osteoblasts just laying down. Here we can see this pink something, that's the osteoid. They synthesize their extracellular matrix, the osteoid. And look at that area. Here we can see they are the osteoids, but these cells are osteocytes. So they were the first onto the surface of this cartilaginous part. So, and they just lay down the osteoid matrix and enclosed by this extracellular matrix. Therefore, they are enclosed by their lacunae. So this is a bony tissue, and that's the cartilage. And gradually, the cartilage, eventually the bony tissue replaces the cartilaginous matrix. So when we're talking about endochondral ossification, that means the cartilage matrix gradually replaced by the bony tissue. So that's the endochondral ossification. But intramembranous and endochondral ossification are just presented during the development. But when the bones are fully matured, we can talk about, and after a kind of remodeling we have continuously in the during the human life, just called oppositional growth. That means, so it's not for the development, it's for the remodeling and for the regeneration after a broken bone. So oppositional growth means underneath the periosteum we have osteoblasts just laying down the osteoid, and at the side of the bone marrow cavity we have osteoclasts digesting the bone tissue, and eventually the work of these two cell population just keep up the thickness of the bony wall. You know, when we're talking about the blood smear, the majority of the cells are red blood cells. They are mature red blood cells, therefore they don't represent nuclei, and they are small kind of discoid structures, and among them we can find, this is an eosinophil granulocyte, which is bilobed. Here we have the two lobes of the nucleus, and the eosinophilic granules are inside, well presented. That is a different one. That is a segmented nucleus. So this is a neutrophil. Also eosinophil. Look at that, that's different. There is a bean-shaped nucleus. So this is a monocyte. Here we can see platelets. So the small somethings are platelets. Eosinophilic. Here we have, there is a lymphocyte. It's the size is really similar to the size of RBCs, but there is a huge nucleus and the thin rim of cytoplasm. So it's a lymphocyte. Now again, eosinophil. That's neutrophil. It seems monocyte because of the bean-shaped. It's really interesting. It's really pinkish, but I think it's just a darkly stained neutrophil because it's not bilobed. It's segmented. It's also segmented, so it's neutrophil, platelets. Now that's nice. This is a bean shape, that's the monocyte. You know, to find basophilic, it's a really hard job because it's just represents the 1% of the population of cellular elements. So eosinophil, neutrophil. That is a lymphocyte because it's quite similar to the size of the RBCs. So neutrophil, eosinophil. No, I don't want to waste the time with to find the, so lymphocytes, the basophil. Okay, now let's move on. You know, also frequently asked story the gland, the acini. So here we have the submandibular gland. It's a mixed gland having serous and mucous acini, but in the submandibular, the serous acini are predominated. We have connective tissue septum dividing the gland into lobes, and from the wall of the lobes we have the small structures called lobules. So let's see them. Here they are. So serous acini. Here we have the serous acini, mucus acini, the foamy cytoplasm. That's the lumen of it. You know, the lumen of the mucus acini are more visible than the serous ones. In the serous acini, the nuclei are round-shaped and well presented. No exact lumen. They are darkly stained in the mucus. Nuclei are pushed to the bottom by the foamy cytoplasm. Just over the mucus acini, we can find some demilons. They are the Januzzi demilons. And inside the serous acini, we can find the zymogen granules. So these dots, these are zymogen granules. And in the mucus, we have mucigen granules. You know, they synthesize saliva and they synthesize enzymes. They appear in inactive form in the acini. And then we should, these glands, these acini should push the, produce saliva toward the lumen of the duct system. So we have the intercalated ducts. This is it. Here we have, it's not a cross section, it's a longitudinal section, but the color is different than the serous acini, and the nuclei are round-shaped. The cytoplasm is pinkish. So this is the lumen of the intercalated duct, but I'm showing a cross section as well. So these big ones are the striated ducts. These are the striated ducts. So these are everywhere. Intercalated are much more smaller. So here, look at that. This is it. That's the proper cross section. This is also. So the intercalated ducts, the size of them are smaller than the acini. We can see the lumen. The nuclei are round-shaped, are at the center. Cytoplasm is pink. So this is an intercalated duct, the first part of the duct system. Then we have the striated ducts. Striated because at the basal part we have really fine striation of the cells having mitochondria because of the really intense absorption and secretion through the wall, the duct wall. And then when the lining epithelium becomes stratified, that is an interlobular duct, but we have much more bigger interlobular ducts as well. So here, they lined by stratified cuboidal, stratified columnar epithelium. Don't mix them up with the vessels. So here we have the flattened cells there with endothelial cells. So this is a vessel. This is a vein. Another interlobular because of the stratified lining epithelium. They are vessels. you know, in the small intestine, the duodenum, we have 3 surface increasing structures. The Kerckring folds. Onto the surface of the Kerckring folds, we have the finger-like villi, the intestinal villi, and the third surface increasing structure structures are the microvilli of the enterocytes. So Kerckring folds are present connective tissue, part of the submucosa, and the villi. Yeah, these are the villi present cell-rich connective tissue, so that's part of the propria. Among the cells, we have goblet cells, and at the bottom of the propria, we have this kind of intestinal glands called crypts of Lieberkühn. So crypts of Lieberkühn, and these structures built up by enterocytes. So these enterocytes, goblet cells, and we have some cells here. That's the one secreting some red granules there into the lumen. They are the Ponnet cells, and we have some cells there collecting their product at the bottom. They are enteroendocrine cells, and maybe look at that. It seems it's after the division, the matrix division. So here we have a differentiating cell as well, and yeah. So enterocytes, goblet cells, Ponnet cells, enteroendocrine cells, and undifferentiating cells are presented in the crypts of Lieberkühn, in the small intestine. Again, enteroendocrine, Ponnet, goblet, and that's the enterocyte. And don't forget, yeah, everywhere we can find lymphoid infiltration having lymphocytes. And don't forget, in the submucosa of the duodenum, we can find Brunner's glands. These glands, these glands are secreting alkalic juice to neutralize the acidic stomach content in the first part of the small intestine. This is the submucosa, and under the propria and separating the propria from the submucosa, here we have the muscularis mucosae layer, which is inner circular outer longitudinal smooth muscle layer. In the submucosa, we can find loose connective tissue having some elements of the enteric nervous system. They are ganglionic cells in the Meissner's plexus. So this is a Meissner plexus. And muscularis externa is presented here. The inner circular outer longitudinal, and between the two layers, we have the Auerbach plexus or mesenteric plexus, also representing ganglionic cells. These are ganglionic cells. And then looking at the surface, if we can find a simple squamous covering, maybe there. Yeah, because it's definitely not. So here we have a thick connective tissue, so it's adventitia. You know, the upper part of the duodenum represents peritoneum. So maybe it's a part of it, because here I can see some flattened nuclei there at the surface, and there is a sharp border. So now we can, you know, the superior part of the duodenum covered by serosa, but the descending inferior horizontal and descending are uncovered. So eventually, obviously, I cannot review all slides, but I try to cover the most important aspects from the basal tissues, the basic tissues, and some of the important and frequently asked stuff related to the GI tract. But this is the exact way of the examination. So the examiner will ask you beside the screen and ask you from the theoretical background, and it's also essential to recognize the structures. Do you have any question?
Sir, I have a question for returning to Hungary. So,
Concerning what?
About starting the practical thing.
Yeah.
So, do we need to return to Hungary like 2 weeks before the practical stuff start, or?
Sunjin, the rector. The thing is, the rector haven't decided yet. So it depends on the exact situation of the pandemic in Hungary. So if we know anything about this story, the rector and the course director will inform you. So the thing is, we're still waiting for the exact decision of the rector, and this depends on the decision of the Hungarian government, so we'll see.
Okay, thank you.
You're welcome. Anything else?